Good morning and welcome to this live broadcast of the third of the four-part webinar series on next-generation sequencing in clinical research. I am Krishnan Alampalam and I am your moderator for today's event. Before we go further, there are a couple of announcements. One is everybody is on a mute except for the organizer and the speaker. This webcast is designed to be interactive and we encourage you to ask questions during the event anytime. You can submit the questions by typing them in the Q&A box which appears on the lower right-hand side of your monitor. We'll try to answer as many questions as we can. If you have any technical problems viewing or hearing the presentation, please let us know by entering it in the questions box. Just to make sure that you can hear me clearly and see the slides, please enter yes or no in the questions box now. Okay, perfect. Thank you. Thank you for your quick response. Now, we are very delighted to bring this educational webinar series on NGS in clinical research. NGS is now increasingly used in clinical research, and therefore there are many questions often. They deal with what to do, how to do, where to start. So in this series, we wanted to bring experts in the field from different continents and share their experience of implementing NGS in clinical settings. I would like to introduce today's speaker, Dr. Clifford Tepper. Dr. Tepper is the Technical Director at the UC Davis Comprehensive Cancer Center, Genomics Shared Resource, and a research faculty member in the Department of Biochemistry and Molecular Medicine at UC Davis School of Medicine. He received his PhD from Department of Pathology at Rutgers Graduate School of Biomedical Sciences and his postdoctoral training in the laboratories of Drs. Michael Selden, Duke University Medical Center, and Dr. Kung from UCD School of Medicine. His graduate and postdoctoral research was centered on cancer genetics and apoptosis regulatory mechanisms. For the last 14 years, Dr. Tepper has focused on defining the molecular genetics of hormonal independence and aggressive behavior of several types of solid tumors, including advanced prostate, breast, bladder, lung, renal, and hematological cancers. More recently, his work focuses on translational applications of genomics, including the optimization of methods for analysis of pathological specimens, the development of clinical next-generation sequencing-based mutation screening tests, and approaches to selection of therapeutic agents. Today, his talk will be focused on the feasibility of using new targeted NGS assay for simultaneous identification of mutations in 160 cancer-related genes in circulating free DNA. I will now turn it over to Dr. Clifford Tepper for his presentation titled Utilization of Next-Generation Sequencing to Identify Clinically Relevant Mutations in Cell-Free Circulating Tumor DNA. Dr. Tepper, please.
Okay. Thank you very much, Krishnan, and good morning, everyone. Thank you for attending. So I'm Cliff Tepper, and I work at the UC Davis Comprehensive Cancer Center, and Krishnan gave a great introduction, and I guess for a little bit additional background to what I do, most of my time is spent as in the Genomic Shared Resource as the technical director, and so we're an NCI designated cancer center, and so we have 8 shared resources that help support the research here, and so the Genomics shared resource was one of the original resources at the Cancer Center, and so while we originally began just doing microarray, primarily on the Affymetrix platform and also custom arrays, now we've kind of expanded, like many of you, to do next generation sequencing and as well as other platforms. And so pretty much right now what also comes along with the sequencing is a fair amount of bioinformatics. So what we try to do is provide just a complete solution to our investigators of any molecular capacity. So if somebody could be a physician with great ideas but no lab space, we'll help them take the samples, collect the samples, and then perform the analyses and perform even the informatics and write up papers. It will be involved as much as they'd like. And but focusing on next generation sequencing, we try to cater to everybody's needs. So we'll perform transcriptome sequencing, epigenomics, whole exome and whole genome sequencing, and now there's a big interest in, of course, targeted sequencing. Because although we can do whole exome and whole genome sequencing, a lot of the people at the center are interested in the actionable genes and therapeutic targets, and so a lot of our work is focused on translational and clinical assay development. And so we have a number of assays that we've developed for either targeted hybrid capture assays, P53 function-based assays, and we've done a fair amount of work with liquid biopsies, a lot of blood expression assays, but today we're going to focus on the cell-free or circulating tumor DNA studies. And so a lot of this is driven by a lot of the personalized cancer therapy or precision medicine efforts at the Cancer Center, and basically that's just tailoring the therapy to patients' specific needs by matching the treatment strategy to molecular features specific for their cancer. And well, this has become, I think, extremely common or popular these days. It has been standard of care for several cancer types, possessing well-defined genomic aberrations. For instance, if there's ERBB2 amplifications, certain point mutations of KRAS, EGFR, and now ALK fusions and BRAF mutations are important. So specific assays have been designed and are FDA approved, for these abnormalities, and then that you can then treat based on that. But a lot of this has been revolutionized by next generation sequencing, because you can rapidly and comprehensively characterize tumors. Whole exome sequencing can find mutations and fusions throughout all of the coding regions, and now pretty cost effectively. And RNA sequencing allows you to do comprehensive gene expression profiling. Now the thing is that tumor tissue is commonly used for these analyses, and that may not always be available. And so blood genomic approaches or liquid biopsies have become of high interest to investigators and clinicians. And basically, when we talk about blood genomics, we'll just say we're doing any kind of genomic analysis based on the blood components, and the information that we're interested in obtaining is going to be focused on molecular characterization of tumors, tumor-specific mutations, or expression of tumor-specific RNA transcripts. And so the analytes of high interest are going to be cell-free nucleic acids, generally cell-free DNA or circulating tumor DNA, What I want to discuss today is exosomal nucleic acids and circulating tumor cells, and also PBMC profiles. And just on the right, this is kind of an example or a cartoon of what you'll find in either whole blood samples or cell-free plasma. And so what we're after is the DNA, and specifically trying to tease out or find the needle in the haystack for mutations in circulating tumor DNA. And so the general assay approaches are quantitative PCR and also, of course, next-generation sequencing with target enrichment either through amplicon sequencing or hybrid capture. And as far as, where is the circulating tumor DNA coming from? It comes from several places. Generally, you're going to find a fair amount of normal DNA in the cell. It can be derived from apoptotic cells or cells that are phagocytized. It can also be coming from autophagic cells. And the green cells here, these are normal, either healthy or inflamed. But what we're looking for in the setting of cancer, of course, is the DNA derived from tumors. And so they'll either be possibly undergoing apoptosis, necrosis, autophagy. And there's recently, in the last couple of years, there's a new form of autophagy or an accompanying feature where the nucleus extrudes pieces of DNA, and the cell will still be alive, so it might actually be throwing that out into the bloodstream. And after a blood draw, that's what we're going to see. We're going to have red blood cells and various sizes of DNA. And there's a nice review by Crowley about this. And so what I think has been of very high interest to us is that blood genomic approaches can address unmet clinical needs in precision medicine. And specifically that you could use blood-based assays, of course, or screens for somatic gene mutations in circulating cell-free DNA. And the nice thing about this is that it's minimally, it can be highly specific, and you can use this for tumor detection and also for monitoring the response of tumors to therapy. And depending upon the technology, you can have various levels of potential for early detection of primary cancers or residual disease following resection, emergence of refractory tumors. And so right now, the exciting thing is that you can use, these blood-based assays as novel molecular diagnostic tools, maybe as companion diagnostics to address these needs. And so for detection and monitoring, these approaches can maybe be tagged on to clinical trials such as MPACT or NCI Match, where you're using precision medicine-based approaches to target the tumors with therapies, but I think we need companion diagnostics to also monitor the response afterwards. And so what we're going to focus on today is a project that several of our oncologists have been doing in pancreatic ductal adenocarcinoma. And so Thomas Semrad is clinical oncologist and works with Philip Mack, who's a molecular oncologist, and also with David Gandara and Lucky Lara on basically the clinical side of this, seeing the patients and treating them. And so this pancreatic ductal carcinoma actually is a good example of a cancer that really needs new methods of assessment. It's the fourth leading cause of cancer death in the United States. Mortality is always rising. It's horrible in that less than 5% of the patients will reach a 5-year survival, 2/3 of the patients die within the first year of diagnosis. Most of the cases are adenocarcinoma, so they're epithelial, derived from the glands, and less than 5% are either neuroendocrine or islet cell carcinoma, and they're particularly rare, and this is the type actually that Steve Jobs had. And so molecular assessment is essential for prognosis and also evaluating candidate predictive markers. And so the thing is, is a very, very challenging cancer because of where it's located. It's intertwined. It's in the abdomen, the pancreas, and it's in the abdomen, intertwined with arteries and veins. It's got ducts going into the small bowel, so it can be difficult to get at. The cancer will usually start in the head of the pancreas, but other problems with getting biopsies is desmoplastic reaction or fibrosis, and also usually when diagnosed it's in an advanced stage, and it's particularly aggressive. And this schematic basically shows that the cancer will start in the duct, and all these red spots are hot spots where it can metastasize to. So there are, because of all the blood supply and the ducts, it's got a number of highways it can take to metastasize to other organs like the liver and the duodenum. And so that's a huge problem with management. And then this in the lower right-hand corner is a nice image showing where you have normal epithelial tissue, and then juxtaposed to that you've got pancreatic cancer beginning, where rather than maybe one layer of epithelial cells, now they're starting to pile up, and it's especially evident over here. And so it's particularly reminiscent of a lot of adenocarcinomas like prostatic carcinoma. And up here you've got some of the molecular events that are associated with pancreatic cancer progression. So KRAS mutation is a major event, telomere shortening, and so these events are characterized. And so thanks to the Cancer Genome Atlas Research Network, a number of the genomic features of PDAC have been defined, and through comprehensive profiling of these tumors and normal tissue from patients. So probably over a couple hundred patients were profiled, and what the studies show that there's 16 significantly mutated genes that were defined. There are common driver mutations like in KRAS, P53, P16, or CDKN2A, and also a number of several other genes that are involved with chromatin modification, like ARID2, and also ARID1A is involved, and also DNA damage repair was significantly mutated, and several other genes involved with other mechanisms. And then when you take all of this data and then do a pan-cancer analysis that some of the informatics groups associated with the study did, they see that pancreatic cancer kind of lies in the middle of these 25 or 27 cancer types that were analyzed in terms of mutation frequency. And so it sort of makes sense because it is a gland, so it's similar to breast and prostate as far as adenocarcinoma, but it's in contrast to, say, some of the other cancers like melanoma, lung carcinoma, bladder that have probably 10 to 100-fold higher mutation frequency, and so that's all I want to say about that. So Now moving to some of the work being done here. So Dr. Semerad headed a phase 2 study of examining the effect of gemcitabine and intermittent erlotinib for pancreatic cancer patients, and collected blood and tissue when possible. And their first interest was to look at the status of KRAS in terms of 7 well-defined mutations. And for this, they used a standard CLIA and FDA-approved assay, the TheraScreen KRAS PCR kit, and I think many of the samples that we've used were KRAS positive. But what they found was that if you can detect KRAS in the circulating tumor DNA, a KRAS mutation, generally that's going to signify that there's going to be decreased progression-free survival and decreased overall survival, dramatic decreases for both. And so this demonstrated the importance of examining KRAS using circulating tumor DNA. And so what we wanted to do with him, he was now interested in basically performing a proof of principle study to demonstrate the feasibility of using next generation sequencing for this and for identifying other mutations in cell-free circulating tumor DNA obtained from these patients. And so what we wanted to do was compare the results of the KRAS mutation analysis of matched tumor and ctDNA samples using NGS to that performed with the CLIA PCR-based assay, the TheraScreen assay, and also to expand the analysis beyond KRAS to identify additional cancer-relevant genes. And so as far as study design, we have tumor and circulating tumors DNA samples that were obtained from the patients enrolled in this phase 2 study, and so there were 28 total patients that we were able to get plasma samples from. At the moment, we're still working on this study. We only have 3 matched pairs that have tumor specimens with FFPE, and so that's one of the one of the hurdles in the study. But there's no problem getting plasma. And so as far as DNA isolations, this was started before we were thinking about even using the QIAGEN system. And so the plasma samples were, the DNA was isolated using the ChemiGen system for PerkinElmer. For FFPE, we used the QIAmDNA FFPE tissue kit. And basically, the workflow is the patients come in, there's blood sampling, centrifugation to separate out the erythrocytes, and then the plasma is taken off and aliquoted and then used for DNA isolation and for analysis of other factors. And as far as the second part of the study design, that was to do targeted amplicon NGS of cancer-relevant genes. And so we're using the FFP tumor samples and the circulating tumor DNA. And for this, we did a couple of runs. We did multiplex sequencing on Illumina sequencing systems. On the MySeq, we did 2x75 base pair, 6 libraries per run. And then we ran everything on the HiSeq 2000 in 2 lanes, 14 libraries per lane. And so then following this, we did the standard sequencing data analysis to focus on identifying somatic mutations. And we have, I'll discuss this pipeline or schema later, but basically we're interested in the alternate allele fraction, the type of mutation, evidence of somatic mutation, and also impact of mutation. as far as for filtering. And then we wanted to compare the results, of course, between the circulating tumor DNA versus FFPE, and to identify other relevant mutations in the samples as well as potential therapeutic targets. And so what we were using, what really interested us at first was the Gene Read Comprehensive Cancer Panel, and here's the list of genes. And we had developed our own panel for hybrid capture, and pretty much doing it independently, we come up with a very similar set of genes. Where many of the genes here are driver genes like AKT, and you'll have your P53 and TSC1 and 2 as tumor suppressors, as well as RB1. So pretty much everything at the time we started this study is covered by this panel. And a lot of the things that were stressed whenever we were starting to go into clinical samples was that whatever we're looking at needs to be actionable. And so, of course, I would love to do whole genome, but I'm brought back into reality when, things have to have have to be druggable to some degree, or have some sort of relevance for maybe subclassification. And so this panel does a very good job of that. Just going a little deeper into what this panel, or some of the characteristics of the panel. It's an amplicon sequencing approach. It's PCR-based enrichment of the target genomic regions. There's close to 8,000 regions or amplicons that are going to be targeted by the panel, and it covers over 745 kilobase pairs of genomic content. And after the target enrichment, it can also be then barcode the sequencing libraries, and so that lets us multiplex everything. And just as an example of the coverage of these amplicons and primer sets, I took the bed file for the assay, and I think it's on the QIAGEN website, and you just upload it into the integrated genome browser or even UCSC. And so here's the schematic of AKT, AL, KRAS. So all these rectangles are exons, and then these exons below that pretty much match every single exon. Those signify regions that the amplicons in the assay cover. So it's nearly 100% coverage. I think unless a region was not PCRable for some reason, it won't be there. But many of the, or I'm going to say most of these regions are covered. And so it's covering all the exons. And as far as the, an overview of the protocol, it comes as a nice kit and a box from QIAGEN, and then our folks in the lab, Ryan and Stephanie, receive it, and then they go to work. And the assay is a 4-tube assay, and we'll add a total of 40. We use a total of 40 nanograms DNA, 10 nanograms per tube, and each of these tubes is highly multiplex PCR amplification for target enrichment, and then library preparation. And everything now uses ampere beads. So so during the assay, it moves quicker. You don't have to use columns. The ampere beads speed up things, and so it's really only a few hours of hands-on work. And as far as a little more details, I discussed this in the previous slide, but this just gives you an example of the primer sets, for each of these, exonic regions. So they're going to be generally slightly overlapping or juxtaposed, so you encompass the entire exon. And that's important, especially when you're doing amplicon sequencing, because you're going to be getting fragments starting and ending at the exact same point. And so you want to make sure there's maybe slight overlap, or they're going to meet, so you can figure things out during analysis. And then the next part of this, after doing the library preparation and then sequencing, is our data analysis pipeline. And we basically, we're focused on identifying somatic mutations, but there's other functional criteria we're interested in. And so, well, we've assembled a pipeline that has several computational tools and software and websites that help us accomplish all of this. And so basically what we want is, after we get the sequence data, we have multiplex sequence data in FASTQ format. That gets demultiplexed, and then we'll do a FASTQC for a quality control. And then we can do a couple of other things too. We can either run this data, we can do for read mapping using Bowtie, and then run it through Genome Analysis Toolkit on our own workstation. Or what we're finding is extremely helpful and rapid is running it through the QIAGEN's GeneRead variant calling pipeline on their web portal. And this, when we've uploaded 28 samples, we have it, depending upon the amount of traffic, we'll definitely have it the next morning, but sometimes within a few hours the analysis is complete, and for our variants. And then when we get the output from the variant calls, we'll either use some custom tools that we have for filtering, and or we'll also get the help from QIAGEN's Ingenuity variant analysis. But basically what we want to do is we want to select for functional mutations, whether they're non-synonymous, missense, stop gain. We want to filter out normal variants, or variants that are occurring in the population commonly, and we also want to prioritize the mutations. So we want to really figure out that, or validate that the mutation is a somatic mutation and has occurred in at least a few tumors. And so we'll get the help from the COSMIC database or TCGA databases, and functional impact with SIFT or Polyphen. And this also is, we'll either do it on our workstation with Genome Analysis Toolkit, or Ingenuity variant analysis accomplishes that too. And we can also visualize the data as well with IGV from the Broad Institute. And so I think I'll just briefly talk about a couple of biological or technical considerations when we're analyzing circulating DNA. And so, the amount, one of the factors that's going to influence what we can detect is the amount of the DNA that's shed into the blood. And so that can depend on tumor burden. Also can depend on the type of tumor or the treatment status. And the other aspect is tumor heterogeneity, which will influence the mutant allele fractions. So just as an example, here are a couple of tumors that we profiled. These are actually melanomas, but when we look at the the mutations and the fraction that alternate allele is present in, you'll see some of these. This is in the tumor itself. This particular one is 17% of the alleles called. So if that was to be, that's 17% in the tumor, so it's heterogeneous. if now we see that being shed into the blood, it's going to be even lower. So when we're doing circulating tumor DNA analysis, we might even expect to see lower amounts of allele fractions for these mutations. The other factor is, of course, the amount of normal DNA in the blood. So there might be more or less depending upon the health of the patient or individual. And also, of course, what's important is fragment size of DNA. And I'll mention that later, but the assay does take this into account. And so if you look at circulating tumor DNA with the bioanalyzer, generally what you're going to see is it's going to be about 140 to 170 base pairs in size. So it is fragmented, at least most of the free DNA in the blood. So that's a consideration. And so you have to make sure that at least you're going to be able to have an assay that will accommodate that. So PCR assay and sequencing, enrichment are all things which can accommodate this. And so our view is, maybe a focus test is more practical, like targeted sequencing. And so here's a workflow that is provided by QIAGEN that we basically follow. So you have the DNA sample. We do a sample QC. I'll jump back at that later. But then if all is good, we go into targeted enrichment, which is a PCR amplification. Then library construction, library QC, sequencing, data analysis, and then interpretation. And below here, all the different tools or kits that are going to be used. So for the targeted enrichment, it's the GeneRead DNA-seq panel kit, and we also of course get the proper library kit to go along with that, and you can put it on any sequencer. And then as far as data analysis, CLC is a good tool. You can use your own if you've got it set up, or if you just want data quickly or results quickly, you can shoot it up to that variant calling pipeline. And variant analysis is a great kind of secondary or tertiary analysis tool. Now, in terms of sample QC, this is very important because we try to quantify the DNA with, well, we'll always quantify the DNA with nanodrop, also with a qubit. Depending upon the amount of single-stranded DNA present, or you'll get varying amounts, you'll get varying concentrations. And so that might influence whether or not things work downstream. So what sample QC does, there's a kit called Quantimise that will basically, it's a PCR assay for a number of amplicons of a couple of different sizes, and so you can run that assay, and it will tell you if the DNA you have will work in the downstream target enrichment steps. And so that's valuable because it not only tells you a yes or a no that it will work, but it will also give you an idea of how many cycles you'll need to do for target enrichment. And so we've seen that some DNA that that we get for this is all plasma derived DNA. Some might only require 19 cycles during target enrichment, while others are going to require 23 or 24 cycles. But this is important. This is important to know and very helpful. And generally, with this assay, you'll, I think when we've looked at, I think generally they've all passed QC with this assay. And what's important is they all end up yielding libraries that can go on this, that will pass library QC, and we'll have enough library and it'll be validated that it can be sequenced, and we get good sequence data from, we've gotten good sequence data from all of our libraries. And so that we're pretty happy about. And so as far as a more detailed workflow, basically this pretty much sums up everything I've talked about. You'll do FFPE, or circulating tumor DNA isolation, and you'll test it with the quantumized kit. You'll do your target enrichment with the gene-read DNA-seq panel that you're interested in using, whether it's a comprehensive panel or a smaller panel that might be more cancer type focused. Then you have your bead purifications with Ampure. Then you'll have library prep and size selection, and then just your enrichment for the libraries, and then sequencing, and then your analysis. And so if you have your own sequencer, your own MySeq, or this can all be done in 4 days. I'm just calling the patients that we have matched circulating tumor DNA with FFPE, I'm just calling them patients 1, 2, and 3. And here I just wanted to give an example of some of the data that comes off. And so this is the data, the FastQ files after they've been mapped with Bowtie. And so now this is the BAM files that we're looking at in IGV. And so what it shows for patient 1 and patient 2, the tumor and ctDNA, we know, we can see here that the tumor does have a KRAS mutation, but I'm just showing a few of the sequence reads here. Each of these bars, whether it's blue or red, if you're not familiar with this, corresponds to either the plus or minus strand of the DNA, and so you're going to get reads from each side. And so whenever you see solid, that means it matches the reference assembly exactly. And then any other letter is a variant. And so we do have a variant here, and this is codon 12 for glycine. And so this is the expected mutation we're looking for. And you can see it's not in all of the reads. But then when we look at the circulating tumor DNA, although I'm just showing a few of the reads, there were over 1,000 reads here. There are no, we cannot detect the variant. So in this sample, at the depth of sequencing we did not detect the variant. But in contrast, if you look at patient 2, there's a fair amount of variants in this codon in the tumor, and we could, although it's at a lower allele fraction, we could detect it in the circulating tumor DNA. So so that just shows that depending upon a couple of factors, we may or may not detect the mutation in circulating tumor DNA, but in many cases we can. And so now we're comparing the results of the sequencing with the panel to the ARMS assay or the TheraScreen assay, or PCR assay. So when this this assay was run in our CLIA lab, the molecular diagnostics lab, and so for patients 1, 2, and 3, they all had mutations detected in the tumor at glycine 12, and also when we did sequencing, whether it was on a MySeq or a HiSeq, slightly different depths depending upon the multiplexing and the amount of reads, we were able to easily detect the mutation as well in the tumor tissue itself. And this is from FFPE, so that sometimes can be a little more challenging than maybe fresh frozen. Now when we take the ARMS assay, and do not think the assay is actually approved, FDA approved for circulating DNA, but it does not detect the mutation in two out of three of the samples. When we look at the sequence-based assay, we actually can detect the mutation in 2 out of 3. It's odd that we thought it was odd that we could actually detect it in these 2 samples that were not detected here, but couldn't detect it in this patient 1. But in any event, it looks like this NGS-based assay at least had the sensitivity to detect this mutation. And so, then we did a little more analysis of the data, because when we called the mutations for these set of results, we're using our defined our filters that are already set for our hard filters that are set for quality calling of variants, whether it's going to be in Genome Analysis Toolkit or the preset filters and quality controls that are set in the GeneRead variant calling portal. So what we next wanted to see is, you do get a file back from the variant calling portal that, in addition to the high confidence calls, you'll also get low confidence and also sites that just weren't called, or very low confidence for variants. And so our hypothesis was that, well, maybe some of the samples that were not being called for a KRAS variant were just because they were low confidence, either in that there were just a very low number of reads. And that actually turned out to be the case. And so, in this one sample, one of the samples that did have that was called for the mutation, 25% of the reads actually had the altered allele. So that's in the circulating DNA. Out of 6,100 reads, we have 1,500 reads with alterations. That's a very high number of variants. And Dr. Semrad had said that this patient had a particularly high tumor burden. So, at least in this case, tumor burden correlated with the amount of mutations that detected in the circulating DNA. Then when we looked at these other samples that we knew had mutations from the ARMS assay of the tumor, you can see there's a variety of alterations. A fair amount. Even though there's 327 alterations found, it's only 6.68% of the reads. And so for some reason, these were actually falling below the thresholds that were for calling high confidence. But in general, to us, we at least think these 3 here, we could be very confident, although it's a low percentage. Maybe for a tumor that might be low, but for circulating DNA, this might actually be acceptable. With these other samples, where we're seeing anywhere from 1 to 12 alterations, that's low, and it's less than 1%. We could do more sequencing, deeper, and get more reads, but it all depends on how we interpret the data, and so we can detect the variants in the circulating DNA. And then when we look at these the samples from these 3 patients and just do some summarizing of the stats for the variants, we see that we can get right here, these numbers here are the number of variants in the circulating tumor DNA that are also found in the tumor. And so we're getting about 50 to 68% of the variants that are in the tumor cold in the circulating tumor DNA as well. And there's, varying numbers. The number or percentage of these variants cold varies with the allele fraction. And then, when we expand the analysis to other genes, for each of the patient, we're comparing or examining the tumor, the circulating tumor DNA, and so we're interested to see, like here, how many of these are actually in common in the tumor itself, and so that's what you're looking at here. And so, in some cases, we detect variants in the tumor that we just don't see in the circulating tumor DNA. And in some cases, we actually detect it in the circulating DNA, but not in the tumor. I don't know why that is. Maybe maybe it's a metastasis and the new clone. The other thing is, I'm not sure if the blood samples were taken at the same exact time as the tumor sample was, so that can be a factor in the differences here too. But you can see that in the tumor, some of these variants are at pretty low frequency, and as well in the circulating tumor DNA. And so I think that'll probably be one of the challenges in the future is how low can we go as far as detecting mutations. What allele fraction would be tolerated or acceptable. And then when we take the results from all of the 160 genes across all of the 28 samples, and what we did was we put all the VCF, the variant call format files, up into Ingenuity variant analysis and did a variety of filtering. And so what you're seeing here is basically the mutation grid for this patient cohort. And so it's highly filtered data. All of these variants had to satisfy a specific allele fraction. They all had to be, they're all somatic mutations, so they are, if any are present in DB-SNP, they have to be pathogenic. And then if they definitely cannot be represented in any of the population databases at they've got to be less than 1%, and also although it's kind of preset based on the panel, they're all going to be cancer relevant. So they're either going to be drivers or therapeutic targets, and what the colors connote are the different levels of allele frequency of the variant. So basically starting with light orange at the lowest, from maybe 0.1 up through, I think the highest here was maybe 0.7. And then what I've done is just kind of chopped it a little bit just so we can focus on the bottom. So some of these tumors, like I showed you in that one of the early figures about the mutation frequency of the pancreatic cancer. It's kind of mid-range for mutation frequency, and so many of these tumors have 0 to 3 somatic actionable mutations, and but some of them, and which might be expected based on the number of genes surveyed. But some of these, like this tumor here, has 17 mutations, and another with 10, and another with 12. And what's interesting is, for this patient cohort, they might be hypermutated, and it's interesting that they're, all three of these samples have a mutation in at least one DNA damaged checkpoint gene. ATM or P53, and also one of the chromatin remodeling or conformation mechanism proteins. So it's just kind of a finding. And so then, what we wanted to do is just what we've been working with is for precision medicine is to come up with rule-based, heuristic based methods for target selection. And because although we, all of these agents, and we're not the only ones, of course, that are trying this. It's great efforts at the Broad Institute. Gad Getz has a beautiful paper on that. But what is a little, I guess, frustrating is we can precisely characterize these tumors for mutations and in actionable genes, but depending upon the paper or study that you read, the success of targeted therapy, I would say at the best is 30%, and will be usually lower. Of course, it depends on the target and the type of mutation you're selecting on. So what we've been trying to do is come up with new ways to select targets, and it's in line with and maybe reminiscent of other approaches, but we'll have our usual technical filters, or the hard filters. We want to read depth of greater than 30, especially when we're doing tumors. We also filter for variant type, like I've discussed, somatic variants that have to be non-synonymous or indels, and then of course the actionable or functional filters. So, do the mutation have a functional impact? Will it be damaging? Will it be activating? And clinical relevance, and is it in a pathway node? So, is it a true driver, or is it in a pathway and maybe very important? And so the other thing we will do when we have the option is we'll also combine it with expression data. And so generally we want to see an expressed mutation. So if we're using cufflinks, we want to have an FBK antibody of at least 10 to give us some confidence that it's at least moderately expressed, or if we can see detect the copy number variant, that'll be important to determining kind of whether it's expressed or completely knocked out. And also allele frequency and tumor composition are important. And so we applied this to some of the data from this pancreatic study. And once we started doing this, of course we didn't use the expression, but once we started applying these rules, definitely our lists get a lot smaller. But we come up with some actionable genes. So here with AKT2 mutation comes up, a good depth in the from sequencing. It's missense, of course. So all of these are going to be missense or frame shift, and the impact judged by SIFT or polyphen is moderate in this case. So so we have some confidence that there might be an effect if we use an AKT inhibitor. And the one thing also that's important is depending upon what kind of drug you're using, agonist or antagonist, is the effect activating or damaging or inactivating? And so that's one of the considerations. And so just to summarize, so it looks like our results demonstrate that it is feasible, and as other people have demonstrated, to use next generation sequencing, especially a targeted panel for identification of mutations in 160 cancer relevant genes, and particularly using this gene read panel. So I didn't show the data, but we got over 93% coverage of all the regions for this assay, and so extremely good results as far as sequencing goes. And then, well, the other finding we have is, or thought is that, well, the specificity of the NGS-based assay is very high. We still need to achieve sensitivity for detection of mutations in circulating cell-free DNA. And so that's going to, you know, what does remain a challenge is when there will be low-frequency alleles or different types of tumors that may not be spitting out a lot of DNA. And so, you know, we're always thinking about how to maybe improve the sensitivity of the assay by various approaches. And so, you know, including deeper sequencing or including maybe mutation-specific primers or probes. And then the other thing is what makes these assays very attractive is that routine or NGS data analysis is now very routine. And so, so I think for the basic analysis, you know, you don't need, especially for an assay like this, you don't need to be a computer guru. You can literally take sequencing data and just upload it, and you'll get your variant calls back. And so I think that's fantastic for the field, and with tools, especially like Ingenuity variant analysis, it makes if you're depending upon your skills with computers, you don't have to have a lot of skills. You don't need command line skills for this. So, you know, so people of varying knowledge bases can get together and look over the data, because I think it's very hard sometimes if you just arbitrarily cut mutation lists at a certain point for whatever reason and then just give it to one person or gives it to another. With kind of tools like this, you can all gather around the monitor or even at different locations and go through the data and discuss the functional relevance. And so, and then in the future, as far as our plans, we want to examine larger sets of matched circulating tumor DNA and FFPE or tumor pairs to, you know, more rigorously evaluate the power of this assay. And then I think people are showing, I think, with, you know, with more optimization, truly the liquid biopsy will be applicable to multiple types of cancer, either for monitoring or surveillance or for, you know, full characterization. And I think with that, I'm going to acknowledge a few people, definitely the UC Davis Comprehensive Cancer Center, and the NCI for providing the CCSG, and our director was Dr. Devere White. Definitely can't forget Dr. Jeff Gregg, who heads our resource, and Ryan Davis and Stephanie Liu, who do all the work in the lab and keep it going. And then, of course, our colleagues in hematology oncology, Tom Semrad, Phil Mack, Irene Hutchins, and Rebecca Sy, who help, you know, organize these clinical trials and patients and patient samples, and our colleagues in pathology and laboratory medicine, Ermi Feldman and Regina Gander-Edwards. And also, I'd like to thank, you know, some of the folks we've been working with at QIAGEN over the, you know, last 3 years, who helped at various levels with Felicity, with kind of optimizing, working with her to optimize the assay, Julie Deschênes, working with Ingenuity variant analysis and clinical insight, and Sean Claremont for making a lot of connections with other people like Krishnan and Raed and Vikram on the other side of the country. And I think with that, I'll thank you. And if you have any questions, I'll be happy to take them now. Thank you.
Thank you very much for the wonderful, wonderful presentation, Clifford. It has been really, really great. I'm sure many of the people, based on the questions I am getting here from the audience, I can very clearly see that they are really excited and they really have a lot of very interesting questions. So,
hopefully not too hard.
No, they're very, very interesting questions. Before I just wanted to also, I know that we are way out of time, but I would like the audience go ahead and ask their questions, and what we will do is we will answer it either via email.
Okay.
Just to justify, just to make sure that at least some of the questions has been asked. One of the first question is interesting. It says, I'm working on oral cancer. We understand that liquid biopsy may be used only for metastasized patients. In case of, so this person is working on oral cancer. In case of first or second stage oral cancer patients, can we get cell-free DNA from blood?
You know, that's a great question. We have several people interested in oral cancers here, and I would think at least it's worth a try. You know, because and actually some of the pancreatic literature I was reading, you know, they were detecting cell-free DNA from the tumor prior to diagnosis, and so from what I understand, with some of these oral cancers, they're very metastatic. So I would think you have a good chance of picking up the circulating tumor DNA, and also depending upon how, you know, whether it's before or after surgery, in case there's still margins, because depending upon how I think the surgery is being done, there might sometimes be margins. It could be hard for somebody to get all of the cells. So those might seed into the vasculature too. So I think it's worth a try to at least, especially if there's some mutations. I forget what the common mutations are, but I think it'd be worth a try to at least, if you can obtain a few blood samples and try it.
Okay, thank you. The next question is, for this targeted sequencing, do you have a paired normal sample, example buffy code DNA, for each patient?
Yeah, that's a great question. For this study, unfortunately, we do not, and so that it's for me, I would love to have normal every time. It would just, or you know, non-malignant. It would just make the variant calling that, you know, for somatic especially, a lot easier, because the rules we're applying when we don't have the normal sample, they're, you know, I think they're consistent with best practices, but I'm not sure I think we might be losing some positives in the mix. So I think if you can get normal, it's great, because also you don't have to sequence to the same depth as the malignant tissue. So you just need really, say, 20 or 30x depth. So you don't need to occupy as much of the sequencing with the normal as you do for the tumor.
Okay, thank you. The next question is, how dependable is the quantumized assay predicting success of downstream steps in the protocol?
Yeah, so we're finding it's very dependable. So I've never run it, but Ryan and Stephanie in the lab have, and they've said everything downstream from that has been successful. So they've gotten libraries. I think the only thing that's, you know, sometimes an issue is, you know, it depends on, you might get different amounts of library, but the quantimise helps you gauge, kind of normalize after even you've quantitated your samples. So it helps everything be a little more consistent downstream. So far it seems like we're getting close to 100% success.
Thank you. There are many, many more questions, so I'm just going to start with one last question.
Okay.
So is this amplicon-based assay designed for known hotspots?
Mm-hmm. Okay. So, the nice, so specifically for hotspots, I think there are a few assays that are being released now, maybe even clinical assays that are designed specifically to the hotspot areas. There are some mutation-specific amplicon assays out there. This assay, although it's not directed at the hotspots, it covers all of them. And so you can easily run the assay, and then if you're just interested in the hotspots, you could just do a quick filtering and select out that data if that's all you're interested in, or your collaborators.
Can I ask a couple of more questions? There are many more here. Do you have the time?
Yeah.
Okay.
Mm-hmm.
Okay. All right. So the next question that comes is, when using deeper sequencing, say 10,000, I think it's so 10,000x for circulatory-free DNA, cell-free DNA, in order to detect low-frequency alleles, how do you identify false positives due to sequencing or PCR error?
Yeah, that's a great question. So I think I don't know the exact answer to that. I think who can help is partly somebody who knows maybe biostatistics well in relation to sequencing, because you just want to make sure that what you're sequencing, you're sequencing individual DNAs, number one, rather than PCR duplicates, and that and so the likelihood of, like in terms of what the error rates are with the sequencer and the particular assay. So I think there's probably ways of doing in the data analysis, and maybe it's even in the variant calling portal that might take into account false positives. And so, but I think, yeah, you probably have to look into that, especially because that's the main concern at the deeper, when you're sequencing deeper. Yeah, that's a great question.
Would you recommend, this is not a question, I'm just asking you this, would you recommend any orthogonal assays to verify any of the, especially for
other assays?
Mm-hmm.
Yeah, so, some people go, for circulating DNA. It's different than, a tumor, because, it's going to, especially if you're at low allele. So, you could design a mutation-specific PCR-based assay, or even mutation-specific sequencing assay that will, that'll, specifically amplify something bearing the mutation. So then that way you can eliminate a false positive as well. Yeah, so I would say some sort of mutation specific assay.
Okay. So based on your experience, what do you, when you publish any of these articles, what do they look for? Are they looking for a couple of methods for verification, or they say, or do different NGS strategies, or two different, or a different orthogonal? Like,
yeah, that's a good question, because the especially if you're using this for clinical work, I think a PCR based. I believe that's what Roach and Ian's lab used when they came out with MyOncoSeq. I think what they would do is maybe sequence not in CLIA, but anything they found that they would think about applying, for maybe a therapeutic. They would then validate in the clinical lab, and I think that was maybe PCR or like Sanger sequencing. And so I think, yeah, so I think, like if you set up an assay that will be designed just to validate the mutation, or maybe just amplify the mutation in a CLIA environment, I think that's a good step.
Okay. With that, I know there are many more questions, but I will just send those questions to you directly, Cliff. With that, I want to thank you, Cliff, for your time today and having a wonderful, wonderful presentation. I'm sure the audience enjoyed it a lot. And this webinar has been recorded, so it will be available to you guys within 48 hours. And also, I just want to remind the viewers that there is one more webinar will be presented next Thursday on the 18th of the month at 6 p.m. Eastern Standard Time. So just it is slightly off time, but this person is from Japan. So again, you'll get an idea of how NGS is being implemented in Japan. With that, thank you very much for attending today, and see you all next week.